Hello everyone, welcome back to the class. In yesterday's class, we learnt about how the insertion of membrane proteins happen inside the ER membrane. And also, we learned different types of transmembrane integral proteins that are what? Type 1, type 2, type 3 and type 4. Type 1 and type 2 and type 3 are single pass transmembrane integral protein. Type 4 was multi-pass transmembrane integral protein. In type 1, the end terminus which is the hydrophilic was in the exoplasmic side, and C terminus, which is hydrophobic, which was on the cytosolic phase. And also we learned about type 2, which was having the end terminus which is hydrophilic towards the cytosolic phase, and C terminus which was hydrophobic, which was present towards the exoplasmic side. We learnt type 3 also, which was similar to the type 2, which is a single pass and having end terminus towards the exoplasmic side and C terminus towards the cytosolic side. And last one, which was type 4, which is a multi-pass transmembrane protein that may have both N and C terminus towards outside or N and C terminus towards inside. We learned this much yesterday, and also we learned how the process of insertion taking place. See here, N terminal starts transfer peptide, which is in blue color, first comes and attaches to the channel. The translocation is happening, or the translation of polypeptide is happening. The pink color one you can see here is a stop transfer site. Stop transfer sequence, when comes in contact with the channel, it stops the channel. The polypeptide which is sufficient gets inside the ER lumen. After that, the N terminal star transfer sequence is cleaved in the N side of the polypeptide. After that, it helps in the insertion of the transmembrane protein into the ER membrane. This is what the process of insertion. We learned this yesterday. Next, we learned type 1, type 2, type 3, type 4 and different examples. Okay. Now we are going into lipid linked membrane proteins. Lipid linked membrane proteins is nothing but the protein, any protein is linked to a lipid molecule. These are exactly like type 1 transmembrane protein, has cleavable sites of start transfer and stop transfer sequence. These proteins are synthesized and initially inserted into the ER membrane. After insertion into the ER membrane, these proteins are transferred to the glycosyl phosphatidyl inositol anchor. See, in previous slide we learned what protein is synthesized and initially inserted. This protein is initially inserted. After insertion, this protein is transferred to the GPI anchor. It is transferred to the GPI anchor. Remember this image. See, in this image you can see there is cleavage happened already. The cleavage of N-terminal start site is cleaved already. After that, the star transfer peptide cleaved protein is released from the channel. The protein is cleaved and transferred to a GPI linked anchor. Here also it gets cleaved from the stop transfer peptide. After that, it is cleaved, it will get and inserted to the GPI anchor. The enzyme transamidase present in the ER membrane cuts the protein free from its membrane bound C terminus and simultaneously attaches the new C terminus to an amino group on the GPI. Which terminus is getting attached to the GPI is, that is C. GPI helps to direct these proteins to the cell membrane. This is how the lipid linked membrane protein or the membrane protein links to the GPI inside the ER membrane. Now we are learning about transport of protein from ER to cis Golgi. So if this is ER, then next is Golgi. Golgi has two structures, cis and trans. This is cis and this is trans. Now we are going to learn how the protein is transferred from the or transported from the ER to the first part of the Golgi, that is cis. The proteins which are to be transferred to the cis Golgi is resident proteins and export proteins. Okay, export proteins are exported from ER to cis Golgi network by following process, that is for following steps: ER specific folding, after that oligomerization, next processing, next transport by formation of transport vesicles, next targeting and fusion of these vesicles. So what are these vesicles? Vesicles are some specialized coated regions of membranes which are helping for the transport of proteins. They are surrounded by coat proteins. Just a minute. They are surrounded by what? They are surrounded by coat proteins covering the cytosolic phase, so that these membranes eventually bud off as a coated vesicle. Prior to fusing with the target membrane, this protein coat is discarded to allow the membranes to fuse directly. There are 3 types of coated vesicles known, each with different type of protein coats. One is Clathrin coated vesicles. Second one is COP1 coated protein transport vesicles, and third one is COP2 coated transport vesicles. All these are different in their protein coat and are formed by reversible polymerization of distinct set of protein subunits. So in addition to coat proteins, various adapter proteins and small GTP binding proteins are required for formation of the coated vesicles. So these are the examples. First one is what? Clathrin, COP1, COP2 coated transport vesicles. First let us learn about Clathrin coated transport vesicles. Clathrin coated transport vesicles are formed they form multiple complexes based on its association with different adapter proteins. See, Clathrin is associated, this type of vesicle is associated with adapter proteins. Adapter proteins 1, adapter protein 3. These are vesicles responsible for the transport of protein from trans-Golgi network to lysosome. Trans-Golgi network to the lysosome. So clathrin, which is associated with adaptor protein 1, adaptor protein 3, are helpful for the transport of proteins from trans-Golgi to the lysosome. And next one is COP1 coated protein. COP1 coated protein is having what? It is having ADP ribosylation factor. ADP ribosylation factor, which is known as ARF1. So to explain this, I'll take one empty slide. The ADP ribosylation, ADP ribosylation 1, for this activation, there is something needed, that is GEF. Remember this GEF, that is guanine nucleotide exchanger factor. So GEF activates the ARF1. Why is this ARF needed? ARF regulates regulates the formation of this vesicle. They recruit coatomer. I'll let you know what is this in a short time. So the GEF will activate ARF1, which in turn regulates vesicle formation by recruiting coatomer. So the function of GEF is to activate, right? But there is one another enemy for this ARF, that is gap. Gap means GTPase activation protein. This will deactivate the ARF. Okay, ARF1. Now we'll see. ADP ribosylation factor 1 is a small GTPase that regulates COP1 vesicle formation by recruiting coatomer, that is a coat promoter. Activation of ARF1 is catalyzed by guanine nucleotide exchanger factor. Deactivation is catalyzed by a GTPase activating protein gap. A lactone antibiotic brefeldin A prevents COP1 coated vesicle formation. It targets the activity of GEF, which catalyzes the activation of ARF. Next is COP2 coated transport vesicles. COP2, and I forgot what is the function of this COP1 coated. So this COP1 coated transport vesicle transports the protein from Golgi to Golgi, means intra-Golgi transport, and also it is helpful in ulta transport, that is retrograde transport, from Golgi back to the ER. Both intra-Golgi transport and retrograde transport is the function of COP1 coated vesicles. COP2 coated vesicles are for anterograde from ER to Golgi. What are they made up of? They are made up of small GTPase, SAR1P, the Seq23/24P complex, and the Seq13/31P complex that sequentially bind on the ER membrane. Budding from ER involves activation of SAR1P GDP to SAR1P GTP by the ER resident protein Seq12P, a guanine nucleotide exchanger factor. Okay, so this is COP2 coated protein. COP2 coated transport vesicles. So how it is made up of? it is made up of small GTPase, that is SAR1P. SAR1P, which is a SEQ23/24P complex, and SEQ13/31P complex. They sequentially bind to the ER membrane. Budding from ER involves SAR1P GDP to GTP, GDP to GTP. Here also this GEF activates the budding from the ER. So here it is showing this overall summary. The clathrine coated vesicle is made up of clathrine and AP1, which help in the transport of proteins from Golgi complex to endosome. Clathrine and AP2, it helps to the transport of proteins from plasma membrane to endosome. COP1, it is made up of COP1 coat proteins. It is helpful in the transport of proteins from Golgi complex to the ER or intra-Golgi complex. COP2, it is a coated vesicle having COP2 coated proteins, where it transports proteins from ER to Golgi, which is an anterograde transport. In next class, we will be learning how the ER resident proteins are often retrieved from the cis Golgi, and what causes for the retrograde transport of proteins back to the ER. Thank you.